All right. Good morning, good evening. On behalf of the Society of the Chinese Bioscientists, the American and the Membrane Trafficking Committee of NIH, I'd like to welcome everyone to today's seminar. I'm pleased to introduce Dr. Wei Guo, professor of the University of Pennsylvania, who will introduce today's seminar speaker.
All right. So it's really my great pleasure, actually, a personal pleasure to introduce Yu Li from Tsinghua University. Li received his PhD from Peking University and did his postdoc work in NIH. In 2008, he joined Tsinghua University as a professor in the School of Life Sciences. So Li discovered autophagic lysosome reformation, ALR, the terminal step of autophagy, and has worked on many other aspects of autophagy. However, more recently, he discovered and named Migrasome, a new organelle for cell-cell communication in migrating cells. Currently, this group is investigating the biogenesis and the physiological roles of Migrasome, which I guess will be the focus of today's talk. So over these years, Li has received many prestigious awards, such as Tanjiazhen Life Science Innovation Award, Chinese Society of Cell Biology Outstanding Accomplishment Award, among many others. I have known Li for many years, both at professional level and personal level. I regard Li as one of the most brilliant cell biologists in China. In fact, I found many of his papers enjoyable to read, as I see his creativity, sound scientific reasoning, and rigor. But moreover, I often see elements of aesthetics in his work. Some of the image and videos are like visual art. So I hope the audience can enjoy his work as I do. So without further ado, Li, we look forward to your talk.
Thank you, Wei, for this nice introduction. Thank you for the invitation. I'm very happy to share our work on the migrasome. So here's the migrasome. As you can see, the cell is migrating. It left a long membrane.
Sorry, Li, can you change the presentation mode to the full screen? Right now, it's just, we see all the...
Okay, let me see. Full screen.
Like the presentation.
Actually,
Yeah, this is the one.
Yeah, that's the one.
Is that work? On my screen is full screen now.
Oh.
We are seeing like all the slides on the side, and also there's
I see. Let me try something. Okay. How about that?
Now.
Nothing changes.
That's strange.
I think you may use
good now.
Oh, now it's good. Now it's good.
Now it's good. Okay,
Yes.
That's strange. Actually, here. Okay.
Oh.
So
that's back.
Yeah.
Okay.
So when the cell is migrating, they left this long membrane tether. Actually, this structure is discovered in the 1960s. The name is the retraction fiber. So on this large vascular grow on the intersection with the tip of the retraction fiber is the migrasome. So briefly, I introduced my background. I have used to work with autophagy in the different aspects of autophagy. So in the 2012, we're just talking about teaching. I started teaching the cell biology class for the graduate student. So I had the chance to read all the classic papers started by the George Plot, the Albert Cloud. So we start to realize actually many cellular structure didn't really get fully explored. So at that time, we started mounting an effort using the electron microscopy as a tool to try to figure out that this structure have been overlooked in the not to be extensive studied. So we found a few. Actually, the most interesting one we found is this. So this is actually the picture started my investigation into the migrasome. So in this cell, actually, we get fixed on the plate and did the TEM. You can see outside the cell, they have many of the debris or the fragment of the cell. But when the EM gets really high quality, you can start to, this is really beautiful structure. A lot of the interluminal vesicle inside this large structure. At that time, because we don't know how to call it, because it really look like the open pomegranate, so we call it a pomegranate-like structure. Actually, we write that in the paper. So my talent student, Liang and Jinya, Liang now is a postdoc in the Randy Schickman's lab. So he figured out the way to purify the PIS and then use like the mass spec to identify the protein rich on the PIS. Then by this way, we get the marker. So he gets at least 100 protein, I told him, you may want to tag the 100, the first top 100 with GFP, then expect to sell. Fortunately, he didn't listen to me. So in the third protein, he found actually, if he tags the GFP in the tetraspin form, you can really nicely label the structure. Then you got this beautiful picture. Of course, with the marker, then immediately we realize that is the migration dependent. So we name that as the Migrasome. So we published the first paper in the 2014, actually online in 2014. So at that time, it really is the description work. We know how it look, we know how it fare, and we have speculation that is for the cell-cell communication. But beyond that, we didn't really know anything yet. So of course, I've been asking for many times what's the difference with the exosome and the other type of the EV. And you can see here, the migrosome is much, much larger. It usually is the diameter of the migrosome is 2 micrometer, where the exosome is 0.5. And the biogenesis pathway is completely different. The exosome is come from the fusion and basically the MVP fusion with plasma membrane then releases intraluminal vesicle, where migrosome is grow on the tip on the retraction fiber. So of course, the most important for us at that time is that rare. That means is that really come from the inside the living organ, neither how it formed, but most important, what is the physiological function. So our strategy both do the mechanism and the physiological functions. Of course, because that is so new, we didn't have any tool. So over the year, we also put a lot of effort to make the tool to start the migrosome. Started from the 2018, which started a very serious the work on the memo. We start to realize the disease relevance. So we try from the discovery to the invention and start to work with the therapeutic. But that is not the really important point of today. So over the year, we have found the probe for the migrosome marker chemical inhibitor. And recently, working with brilliant engineering in the Tsinghua University, we developed several the image method and the microscopy for the migrosome observation. So this is one good recent example with the light field microscopy we built and the adenosine method we developed together. So by the work with this engineer at the beginning of the project, really gave us a lot of good idea how to further look deep into the in vivo image. Now, we already know, that is really the real stuff. Now we know, the migrosome actually is occurring many places. So this is zebrafish, the embryo. As you can see during gastrulation, a lot of the migrosome can form. And this is for the neutrophil, that is the mouse liver. If we take the in vivo image, you really can see a lot of the migrosome formation on the inside the bloodstream. And this is circulating the tumor cell, which is making is moving where generates the migrosome. So the fate of the migrosome have two. So some of the migrosome, if, for example, it's formed on the top of the macrophage, it will be phagocytosis quickly. But in many cases, actually, once they form with the migrosome, they can stick on the extracellular matrix or on the top of another cell. So they just stay there, become stationary. So for this, we found the 3 modes of the function for the migrosome so far. So the first one is the lateral transfer of the material. So this is one of my most interesting movie. As you can see, the green cell actually is producing migrosome. Then the red cell is coming and eating it up. During this process, all the material in the green migrosome will transfer inside the red cell. So inside this, the migrosome, there could be the lipid, protein, and very surprisingly, we found RNA. Actually, this is not just any random RNA. This is the full the length mRNA. So actually, we found once the migrosome be eaten up by the recipient cell, this mRNA somehow can escape from the endosome, the lysosome, get into cytosol and translate it into protein, and then modify the recipient cells the behavior. So of course, this study, the shortcoming of this study is we only demonstrate that can occur in vitro. Now we are working why the mRNA also can transfer between cells in vivo. For example, during the infection, you have the neutrophil, while the neutrophil migrosome mRNA can get into for example, macrophage, and lend this all the encoded enzyme activity to the macrophage. So the second the mechanism we found is the organelle homeostasis. So that is really surprising finding. Many years ago, I used to work with autophagy, so I'm very familiar with the idea of the mitophage. When the mitochondria is under damage, this damaged mitochondria can be degraded. So actually, in our study, we found when the cell undergoes very mild the mitochondrial damage, the damaged mitochondria can be actively transported to the migrosome. And you can see the red panel, a lot of this damaged mitochondria is accumulating inside the mitochondria. We name this process as mitocytosis. Basically, eventually we show this is really important for maintain the neutrophils homeostasis. So for the neutrophil, when it's actually is matured in the bone marrow, when it start to get in the circulation systems, start with a long distance migration. If we knock out the gene required for the migrosome formation, so this neutrophil will have very short half-life because all the damaged mitochondria build up, eventually killing this cell. So it really makes a lot of sense if you think about it, because for the migration, you really need a lot of ATP. That means mitochondria will work a little bit harder. The more RS will be generated, then accumulate all this respiration-related toxicity. Then you balance with the mitochondria-dependent mitochondrial homeostasis pathway, so everything becomes even again. So so far we're only in that paper, we only talk about the one aspect of the mitocytosis, which is the neutrophil using this mechanism get rid of the damaged mitochondria. But put into the the larger aspect of the immunology, because we know the mitochondrial DNA is very potent induced for the innate immunity. So now we are actively working on whether this could be the part of the immune response process, where the one cell generates this, we call this mitosome, can be get into another cell and induce the C-gas pathway. But so far, the most important function of the migrosome will be the really targeted delivery of the single molecule. Very early on, we found the migrosome highly enriched with the single molecule. So as you can see, this is the zebrafish gastrula cell. So the red color is CXL12, one of the very important chemical. So our mass spec analysis show migrosome actually is enriched with the chemical, cytokine, morphogen, and growth factors. And just from this CXL12, you can see how much CXL12 is enriched inside the migrosome. Why that is important? Because for many biological processes like immune response, like the development, you really need a different type of cell, know where they go, what they want to do, and what's the time to go. So you really have the mechanism to put the spatial information, temporal information, and the specific chemical information together. So we think like the migrosome is one of these mechanisms doing this such a job. So if that is the case, then migrosome probably will play a role in the embryonic development, in the tissue regeneration, immune response, and angiogenesis. And in the pathological condition like the tumor metastasis, probably migrosome also will play a role. So the first case, like we're starting with migrosome. Actually, this is the first like the physiological function of the migrosome, is we are using zebrafish embryo. The reason at that time is we didn't have any other method to really observing migrosome in the mammal. So for the zebrafish embryo, it's like translucent, like it's very easy, it's great for the image. And during the gastrulation stage of the zebrafish embryonic development, almost every cell is moving. So we almost immediately see a lot of the migrosome inside the gastrula. And actually you can say the migrosome is generated and then it's like detached from the cell. Eventually all this, so once we isolate enough the migrosome to the mass spec, we find CX12 is highly enriched in this like the migrosome. Eventually we are able to put into the models to find actually for many cells actually they're generating the migrosome. They are like accumulate the cavity on the, I'm not sure that is working, but anyway, like a cavity underneath the embryonic shield. Then as that became the regional queue, using the CXL12 attract the group of cells called the dorsal forerunner cell. Eventually, this becomes determine the left-right asymmetry of the zebrafish. So since it's a published paper, I wouldn't like get into more detail. But the bottom line is like using the migrosome, like the embryo is able to build up this like the region like the developmental Q center. Use this like the center, they make the gradients like they are able to build the more complicated gradient. So the second case like with this is also in the context of the developmental biology. I'm always fascinating with like the pattern formation, like why the blood vessel looking at this pattern but not growing into another pattern. So like the angiogenesis, the biology of angiogenesis is well understood. You have VEGF, which is like the pro-angiogenic factor, which can signal to the tip cell and guide the tip cell's proliferation and the migration. So for us, we use this chicken like the embryo model, CAM model, because like in a few days, you have the really robust growth of the capillary formation. And also again, that is we started this project at very early of the time. So we are not able to like observing like the mammal at that time. So the chicken embryo is a great system. You can take it ex vivo and you can like really put open the window in the egg to watch everything. What's really after a long time like struggle is take like more than 6 years to finish this project. Actually, we found something really interesting. So this blue cell, this blue image you can say is monocyte. So in the normal time, this monocyte just sitting inside the blood vessel. But there's the angiogenesis. This like the monocyte will be rushed out to the blood vessel, and when they're migrating, they left a lot of the migrating track. And on the migrating track, they left many migrasomes. Inside the migrosome, if we purify use like the quantitative like the mass spec, again we find like the angiogenesis factor like the VEGF-A is highly enriched. So I like to point out this is equal protein loading. You can see how much enrichment of the VEGF in migrosome based like in the cell body. So eventually we were able like to put out the vanguard model of angiogenesis. In this model, like the mobile cell, like the monocyte, can really run on the like the CAM, left track of the migrosome. Then like the tip cell will sense this like this cue and follow with like the monocyte's migration track. So we think that could be a very neat like the pattern formation mechanism. So now we are like try to work out whether other pattern, for example, like the could be follow the similar like the principle. So far, like so we have did a lot with like the web. Our work is mostly in the development context, but since like I did my postdoc, spent 6 years in the Dr. Michael Leonardo's lab as an immunologist. Of course, like for the adult animal, most of the cell actually is moving, is the immunocell. So like now my lab is come back to immunology, like so we work all this like the cells listed here. I give you a few examples. You can see neutrophil will produce a lot of the migrosome inside the blood vessel, and the monocyte is after LPS stimulation produce a lot of migrosome. NK cell, T cell, eosinophil. I just give you a few examples of them, like almost all type of the immune cell after stimulation, they will generate migrosome. So now, like with work, like we have the hypothesis, like for example, now we know the cell is autonomous, which is like about like 10 micrometer, and the major effect of this cell, like is the cytokine. They secrete out, it gets diffused into the bloodstream or in any microenvironment. There are 5 to 10 nanometers they are passing. So we think the migrosome is this sort of semi-autonomous unit, like which is around like the 1 micrometer. And we think like they can do a lot of them, like for example, package a lot of the cytokine inside the 1 migrosome. And then they have the combinatorial signaling, like they do a lot of things, like so we don't think like the cytokine alone can be done. So my lab is almost like the majority of my lab explore with this aspect. So now come back to with the mechanism. So like really for figure out the gene to study the migrosome, we use educated guess screening, and sometimes like the log you always have. So now we find migrosome biogenesis is a super complicated process. I can divide it into cellular as a 3-stage: nucleation, maturation, and expansion. So first, I'd like to introduce a term like the migrosome formation site. As you can see here, like this green dot is labeled by the PI45P2, which is before the migrosome formation. You already see a lot of synchronization happen on this like migrosome formation site. So with operation defined this formation site in 2017, we'll find actually integrin is absolutely required for migrosome formation. And integrin will form this very small like the puncta before the migrosome formation on this like the intersection of the retraction fiber. So like over the year, the first project we carry out is like using the screening. So basically we take this logic. If that's a gene, a protein is important for a machinery important for migrosome formation, very likely it will be localized on the top of the migrosome. So what we do is we purify the migrosome, do the mass spec, and use this candidate to do the like the SHRNA screening. And eventually we are looking for this like the candidate gene. They knocking out didn't affect the migration of the cell, but specifically affect the migrosome formation. So actually we found that like the most of the gene is clotting into the glycosphingolipid, like the metabolism. Then of course let's come to the lipid. So we like collect enough of the migrosome and carry out the quantitative like the lipidomic analysis. That's like a really horrible experiment to do. So for every experiment, we need like 400 dish of the cell to make enough like the migrosome to do the lipidomic. So the data is very clear. So the sphinx lipid is highly enriched inside the migrosome compared to the plasma membrane or compared to the cell body. So like I don't know whether you can see here. Then we use this like among them the sphygmomyelin is highly enriched. Then like we use this like the protein probe, which is purified from the earth ore. It's a no toxic lysine, like which can bind into the clustered sphygmomyelin, as you can see. So sphygmomyelin is highly enriched in the migrosome. And if we're using that as like the movie, like you can see, you can appreciate like actually the anti-lysine signal is increasing during the migrosome formation. That means this probably is likely is a de novo synthesized. So to test whether the sphygmomyelin is required for migrosome formation, we just injected the sphygmomyelin hydrolase into the culture and do the imaging. And as you can see here, like once the sphygmomyelin is is injected, like all the migrosome will be collapsed. I'm not sure whether you can see here. Oh, okay. Could you see the imaging? Okay. As you can see, the green color is gone, and then the red, which is the migrosome, will be collapsed. So sphygmomyelin is like is converted from ceramide from the sphygmomyelin synthesized. They have the two, one is in the like the Golgi, another in the plasma membrane. So we eventually figure out that sphygmomyelin synthesized two, which did the job is required for the migrosome formation. I wouldn't get into all this detailed experiment, but let me tell you this. So if you knocking down the sphygmomyelin synthesized to migrosome blocking, formation will be blocked. If you adding the sphygmomyelin back, you can rescue this effect. But the most surprising thing is this. So when we're adding, conjugate the GLP on the sphygmomyelin synthesize, what we find is really amazing. So as you can see, the sphygmomyelin synthesized were forming the small puncta on the leading edge of the cell migrating. So when the cell, then they become the binding to the extracellular matrix, become immobile. When cell migrating away, then this sphygmomyelin synthesize grow into the migrosome. So with this, like so we think like the assembly of the sphygmomyelin synthesized puncta actually defines the nucleation stage. So the implication of this study is the migrosome formation actually be predetermined hours before, and migrosome form actually on the leading edge of the cell, not in the trailing edge as I always thought. So the second stage is the maturation stage. So because this is by guess, because almost like from my experience with cell biology with autophagy, almost every membrane does probably phosphoinositol were involved. So at the beginning, we started screening which phosphoinositol getting enriched. So we labeled the cell with like the pH domain. Very quickly we found actually pH domain like the PR45P2 is highly enriched on the migrosome formation side and on the migrosome. Then the PR45P2 is converted by the PIP5K1A from the PR4P. So we found actually the end of PR45K1A will be recruited to the migrosome formation side also before the migrosome actually start to form. So basically this PR45P2 is then de novo synthesized at there. So once they forming the PR45P2, we just did a very quick screening for all the PR45P2 binding protein. We found actually ROB35, which is before it's diffused on the retraction fiber. Once the PR45P2 gets generated, it will be recruited on the migrosome formation side, eventually driving migrosome formation. So I will skip it because it's published data. I will have skipped all the detail by telling you this. Eventually, one of the functions how the ROB35 is by binding and concentrating the integrin to the migrosome formation site, eventually provides the anchor point. So actually the expansion stage is something really interesting, which is we figure out like that is the first mechanism we correct. So this is purely by luck. So as I mentioned earlier, like the first gene we use as labels migrosome is tetraspanin 4. You have this beautiful like structure. Very soon we realize when we overexpress tetraspanin 4, actually you can dramatically induce migrosome formation. So like really is like for some cell, if you don't, they can migrating, they didn't produce like the migrosome. You just always protect tetraspanin 4. You have a lot of migrosome 4. So tetraspanin is a large protein family, has 33 members. They all have the 4 like the transmembrane domain. So a lot of important like the protein, like important for the immunology and is like to belong to this family. So as you can see here, like when migrosome is growing, actually you have the more and more tetraspanin getting recruited on the surface of the migrosome. So that means that is like something is actively recruiting. There are 33 like the tetraspanin. We found 14 of them when you overexpression, you can induce migrosome. If you finding one of these like the migrosome promoting tetraspanin, if you knock out this tetraspanin in any cell expressing this cell, then you can reduce migrosome formation. But this is like the most dramatic evidence come from this experiment. So basically, we purify the tetraspanin as a recombinant protein. We insert into the GUV. Then we use 2 different methods, use a mechanic force, transforming the GUV into the long membrane tether. In presence of the migrosome, tetraspanin, the migrosome-like structure will automatically form. The reason is like eventually we figure out is tetraspanin were like the organized were into this like the wrapped like the microdomain called tetraspanin enriched microdomain. Inside is highly enriched with cholesterol and other protein like integrins. So when that is happened, basically the migrosome formation process is thousands, hundreds and thousands of this like the microdomain with the diameter as like the 10 nanometer. We're assembling into this like the micrometer scale like the we call it the tetraspanin enriched microdomain. So once they become the microdomain, go through the purely like physics process. So in this project, it's really fun to work with my good friend Michael Kozlov. He is like the biophysics in the Tsinghua University. Basically, he built up the physics model to explain why you have this microdomain assembly, then you have the bulge out of the migrosome-like structure. So I wouldn't get into mathematics. I admit I didn't really understand, but like that is how the Misha tried to explain to me. If you think about like you have the rubber band, the sum of the section of the rubber band is very soft. Like some section of is very rigid. If you pulling this rubber band, the soft part becomes thinner. The rigid part will like resistant to the thinner, so that will be bulged out. Then if you adding, you have the assembly of the multiple section of this like the rigid part together, then you will have the glow of this migrosome. Eventually, that is exactly how the like the Misha's like the prediction. So his model, his like the theoretical model predict migrosome will be much rigid compared to other member, for example, that's retraction fiber. Then we did use the atomic force like the microscopy to test this hypothesis, then verify his theory. So eventually we are able to put this like the model, which is we call like the membrane stiffening model for the migrosome formation. So in this sense, if we put everything on what I told you, it's really like the biochemistry reaction driven. So all the catalysts before the migrosome formation, a lot of them have been done there. Then eventually, really the bulging point is the biophysics, is the physics. So this study also gave us another like the project, which is not directly into linked to the migrosome, but is relative to the property of the tetraspanin-rich microdomain. So very early on, we noticed something really interesting. So in the left panel, all this red is just dye. As you can see, like the migrosome suddenly become permeable. The dye can get into the migrosome, but migrosome still kept like the integrity. This completely didn't make sense. Like we know when you have the membrane vesicle, once it starts to rupture, you will have the become permeable. It should be immediately rupture. But with the presence of tetraspanin, this didn't happen. So this project like promoted, we get into another like the project, eventually realized whenever you have the damage, membrane damage on the cell surface, very quickly, the tetraspanin-enriched microdomain will be assembled along the edge injures like the site to basically make them almost like a bank surrounding the damaged site. Once it's set up, because it's very rigid, it prevents the damaged sites continue to rupture. Then eventually allow the other mechanism, for example, the escort mediates the membrane repair to repair the membrane. So by the way, we know he's like independent, he's a professor in the Tsinghua University. He's still working on the tetraspanin and the migrosome. So put everything I told you together, I think for me is really surprised. At the beginning of my study, we think the migrosome is just they're migrating away, something really sticky be left behind. Now we actually uncover this the very complicated process, very programmed, the biogenesis process. Now our lab is carry out the genome-wide screening. So far more than 30 genes have been identified. We still didn't really know how this gene work, but it look like we just the tip review the tip of the iceberg. So in the final few minutes, I really want to talk about the unpublished work. We really want to talk about why this signaling molecule, cytokine, chemokine, are getting enriched in the migrosome. So this is the original the picture I observed. At that time, I'm very surprised why you have this little intracellular vesicle, luminal vesicle accumulate. What's this vesicle? Where they come from? So we know this is the clock work by the secretion, by the generation of the scientists. So basically, all the secreted protein were made inside the ER. For this audience, I probably didn't need to explain. Then they come from the ER as the COP2 vesicle get into Golgi, eventually through the different type of the secretion carrier, they will be the fuse with the plasma membrane and the exercise out. So the cytokine secretion basically the same logic. So just my student who also the Haifeng, he also work with this mitocytosis paper, realized the striking similarity between the neurons, the axon terminal, and the migrosome. Basically, this is all is the unit transporting for the neurons axons. All the the neurotransmitter have put into this the presynapse vesicle, direction transporting to the axon terminal and release there. We find the action now basically the migrosome follow the same logic. So the key difference is in the migrosome, in the retractor fiber, you didn't really have a very long the microtube, but you have a lot of the bundled actin, as we can show here. And on this actin, you have a lot of the vesicle. So of course, we focus our study on the what kind of motor protein is driving the movement. So when we're talking about the actin, then we know we're probably talking about the myosin. That's exactly the case, as you can see here. If you put the myosin 5, you can see it's highly enriched in the migrosome. And so this is the image is taken by the GI SIM, developed by the Professor Dongli in the Institute of Biophysics. So basically, this technique can give us a single molecule resolution. As you can see here, the single vesicle of the myosin 5 can be transporting this and accumulating inside the migrosome. So this is really myosin driving active transportation. So now from the great work it's like the fusion part of the media based by the SNAP proteins. So again, here we take the one of this base SNAP VAMP2 on the migrosome. As you can appreciate from this, at the beginning, VAMP2 is as a little vesicle inside the migrosome, but eventually, once they fuse, they become part of the membrane. That means actually during the migrosome growth, this intraluminal vesicle is start to fusion with the membrane, and this vesicle is come from the cell by the transporting by the myosin 5. And again, similar to this process also is regulated by the calcium. So the left panel, as you can see here, with a lot of active calcium activity. In the right panel, if we put the TNF, which is one of the cargo, the secretion molecule, with GLP and the always present cell. If we're adding the calcium, blocker, you can say all this the TNF cannot release from the migrosome. Then many of them just get accumulate inside the migrosome. So the same thing is happening in vivo and in the immune cell. Now we are able to show so this small vesicle inside the migrosome actually is the classic the secret carrier. So that first generate from the cell, follow all the classic secretion pathway. But when the cell get into migrating, the exocytosis pattern is changed. The reason we still don't know. But all this secret carrier will be redirected to the rear end of the cell. And it's there, they binding to the myosin the 5 and directly transporting it to the migrosome and regulated by the calcium and the fusion and the release. So that means actually, that is the mechanism for the localized secretion for all these secretory proteins. So this actually happens in vivo. So this is the imaging actually we take a lot of time to set it up. So basically we can show the active monocyte can deposit this TNF loaded migrosome inside the blood vessel. So of course with this, I think the probably have a lot of if an immunologist quite an interesting implication for the immunology. So now my lab is work with a few idea how this the migrosome bond the cytokine can play different role in the immune response. So it's now it's more than 10 years I start with a single the electron microscopy the picture. Now we're very happy the more and more people start to start work on the migrosome and get really beautiful work done. We have the first the migrosome meeting this last year in Shenzhen, sponsored by the SMART, the Shenzhen the medical Academy of Research and Translation. So finally thanks to all my students to work with this project and for all my wonderful collaborate. Thank you. I'd like to take the question. Yeah.
Well, thank you. Very beautiful talk. And so for the audience, if you have any questions, please put your questions in the chat box and we will actually lead the discussion.
Okay. So to be selfish, I have a few questions actually, but let's see. I'll try to save a lot of time to other people. I saw. Well, why don't I read first? Okay, from XC. Aside from migrating blood cells, in what other organ systems or cell types do migrosome appear? If present, what are their associated biological functions?
Yeah, I think that's really the question we try to understand. Now, I tend to think this way, what cell can migrating during the embryonic development, most of cell can migrating, right? And then in the adult animal, so most of migrating cell is like the immune cell. So of course, the immunology is a part of. But during the scenarios such as like the tissue regeneration and damage repair, fibroblasts will be very highly migratory. And during the like the EMT for the metastasis, even the epithelial cell origin cell can be like become migration. So I would think like all this sort of the like the scenario will be probably will associated with the migrosome formation, and probably the migrosome will play some role inside this process.
So from why, from that young, great talk, would the migrosome be washed away during cell culture digestion?
Yes, like actually migrosome very sticky. So if you really want like the purify migrosome, one good place to check is we publish in the current protocol with like the migrosome like the purification procedure. Also, you can find in my lab's like the website. So what we usually do is like we first put a little bit of trypsin, remove the cell, and then we further remove the migrosome. Because the migrosome enriched with a lot of integrins, so it's very sticky.
Yeah. Okay. From Bing Bing to everyone, thank you for the great talk, Dr. Yu. Besides live cell imaging, are there any other tools to visualize the migrosome activity in the in vivo tissues?
Yeah, so now we are really trying to like establish a method for the purification and the quantification of the migrosome. I think it can be done in the body fluid like the urine and the blood, but now we still have the difficulty to purify the high purity migrosome from the tissues. So I think it's durable, but we still don't get that yet. Now, because like the in vivo imaging, I think that I put a lot of the restriction for it's really required very sophisticated like the image apparatus setup. So I really want to develop some like easy way to detection and like the analysis migrosome.
Okay, from Zhang Sheng, is migrosome always passive, leaving behind, or they have a pioneer role in the front end of the migrating cells?
Actually, I used to think like the after living cell had nothing to do with the cell generation with the migrosome, but I'm not so sure now. So I think like I really don't know. Yeah, because one thing, for example, we find like many receptor can enrich on the migrosome, can be shedding from the migrosome. So in some type of cell, that means the receptor on the cell will be reduced. So the cell migrosome actually in this process is changing a lot of its our behavior. But still it's some very early observation. We didn't really like to have the concrete like the evidence to support it.
Okay, for a question from Yihong, do you see protein aggregates in migrosome? If they can be used to get rid of damaged mitochondria, can they be used to get rid of bad proteins? Good question.
Yes. I think in some cases we see this, and one of my colleagues is studying this. Yeah.
Okay. From Zheng Wei, how sphingomyelin lipid synthesis is initiated and regulated during the migrosome formation?
Which I don't know. We think this like assembly of this beautiful like the sphingomyelin synthesized punctile. So now we are working with. We will think like that probably be post-translation modified. And I mean, that definitely have a lot of the biochemistry behind this. So we're still working on this.
Okay. And a question from Yung Li. How do substrates on which cell migrate affect migrosome formation?
Yeah, so like so we published the paper a couple of years in 2017, I think, like we found like basically you really need to have the matched like the integrin with matched ECM to have to produce the migrosome. So basically, any type of ECM can support migrosome formation as long as you have the expression of this like the matched integrin subtype.
Okay. A question from Marianne. Do you see any chaperone heat shock proteins in migrosome?
Yes, quite a lot. Yeah, but we don't know why they are there. Yeah.
Okay. A question from Yihong. How are certain vesicles sorted into migrosome? Is there any sorting mechanism or things?
They have to be, but I don't know yet. So we just make this observation, which we absolutely have no idea how it gets sorted.
I think it's a good question. Yeah. Also, from John Hammer, how does varying ECM density affect the extent of migrosome formation? What fraction of the cell's volume in shed per minute? It's a good question.
Could you repeat the question again?
Yeah, how do varying different ECM density affect the extent of migrosome formation? And what fraction of the cell's volume in shed per minute? It's a very
for the first, so very good question. For the first question, I think in the 2007 paper, we did a sort of very rough experiment. Basically, by coding the different concentration of the fibronectin, we know like it's basically sort of this bar shape. If you code too little, it wouldn't like induce too much migrosome formation. But if you coding too many, also it will suppress migrosome formation because that didn't like the migrating at that time. So I think like we didn't have really like the fun like this like the mathematics to how much, but we know that it have the density definitely is matter. The second question is like we didn't I didn't calculate, but that's a good question. I should do that.
And here's a good question. How long will migrosome in the blood circulation last if they're not eaten by other cells? And will they eventually burst and release their content into blood or tissues? I think it's a very good question.
Yeah, that's a really good question. We try to figure out. It's not like straightforward as it looked. For example, like if we compute a migrosome and injection back to the blood vessel and to check how much like this migrosome were kept in circulating. But in this case, like we found like most of the migrosome will very quickly be engulfed by the Kupffer cell in the liver. So now we'll try to figure out the chemical biology way to monitor the migrosome directly generates in the blood vessel, what they like the lifetime is. So we're still doing that. But that is a really good question.
Yeah, and relate to that, may I insert a question? I mean, you're looking at the VGF and CS12, or I forgot the number. Their topology is actually, there should be secreted out. Their topology, I mean, if they are eaten by macrophage or something, it should be inside now. But their functionality actually was exerted from outside.
Yeah, yes, this is the very confusing part. So let me show you this like the picture. So I think it's really important to remember there are two phases of them. We see a lot. For example, in the chicken, like the embryo, and in the zebra embryo, once migrosome be generated, they were sticking to their place. They are not be eaten by other cell. So then they have enough time. All these secreted protein cytokine or the morphogens inside this intracellular vesicle were fused with the migrosome membrane, slowly releasing out. Then there's like a soluble protein binding to the receptor to affect. But then in another case, for example, if you have the fibroblast around or you have the macrophage around, they are eating that up. So I think this is presented. So for this once eaten up, all the separate protein, just another protein to be digested. But some of the enzyme, some of the mRNA somehow can figure out, we still don't know how they do that. They are usually be phagocytosis or be endocytosis, but they definitely can find the way, escape from the lysosome and start to change the biochemical property of the recipient cell.
And this question also deal with the synthesis of the vesicles. Where are those vesicles come from? I mean, they're synthesized from the cell body, like a soma-like structure from an equivalent of
Yes.
Or but also in your sphingolipid paper, you actually show local de novo generation. So what's
so if you look at the migrosome, I think that for the migrosome, the surface, let's say the membrane of the migrosome, for this lipid, many of the lipids generate de novo.
I see.
For example, sphingomyelin and the PR45P2. And then probably they also have the lateral transport of the lipid. For example, migrosome is highly enriched with cholesterol. This probably come from the lateral transport diffusion of the cholesterol within the tetraspin enriched domain. But then you have another source of the material. This like the intraluminal vesicle will be transported actively, driven by the motor protein myosin 5 from the soma into the migrosome.
Okay, got it. Yeah. So another question from Zhang Shen. Is there any specificity of cargo inside of the microsome? How can the cargoes being sorted into microsome? Is there active?
Really good question. I don't know yet.
Yeah,
we're working on that, but we absolutely didn't have any clue yet.
Okay.
So for example, for the damaged the mitochondrial paper was mitocytosis paper was published. Only damaged mitochondria get it.
Yeah.
And we explained in that paper is like the damaged mitochondria have the higher affinity with the outward microtubule, like the motor protein K5B, have the less affinity with the dynein. But why damaged mitochondria have we still don't have the idea.
Yeah, talk about mitochondria. And do you see the recipient cell, the cell internalized microsome have a higher level of STING?
Yes, we did experiment, but the data is negative. Yeah.
Okay.
So we think that we probably didn't look at the right like the biological context to this experiment.
Okay. Yeah. Another question from John. Is the polarity of actin in the microsome mixed? I bet it is. John said. I didn't bet.
Yes. So I don't know.
Okay.
Because you really can see the myosin is like the really beautifully like transporting the like unidirectionally. So I always think like probably this actin bundle is polarized to the one direction. But again, I cannot rule out in some cases you have the mixed, then you are allowed to like the biodirectional transporting. Yeah, this is all very good question, but I didn't really thought about it yet.
A question from Hong Yu. What is the advantage of a microsome mediated recreation versus exocytosis? Does is specialized in
So like I always think there are several like advantage. The first one it's really allow the site of the localized exocytosis. So basically, it's not like in any surface of the cell you can secrete. You only can secrete it in the microsome, which is far away from the cell. So I would say like that probably can avoid this like the cytokines to come to act on themselves. The second thing is like actually we know like the different kind of cytokine could be packaged into the one microsome and released at the same time. So that means like you really have this synergetic. So this idea called the combinatory signaling. The idea is, for example, you have the inflammation, you really want the IL-6 and the TNF-alpha work together. This is 1 + 1, but it's actually larger than 2, this sort of effect. Then the second thing is, if you think about, for example, in the bloodstream, all these like the cytokine once released will be very quickly diffused. But you put into the microsome, you make them like, let's say, sticky to the certain inflammatory site, allow them by the through the SNAP mediated fusion slowly release this stuff. You really can build a high local concentration over there. So I think that is basically another way to build up the local gradient.
Yeah. Here's a comment from Josh Zimmerberg. Sorry, Josh. I tried to paste the picture, but see JEV for EM or for fusion pore for MCMV, which seems to be distinctive from Migrasome. It has much larger internal organelles and but directly from the cell membrane. It's a good comment. Yeah. So,
yeah. So what's the structure called?
MCMV.
MCMV. Yeah. That is quite. So I think the Migrasome really have a lot of the selectivity, because you everything getting to have be transporting through the retraction fiber. There's some sort of sorting mechanism. Whether the MCMV is really directly bound, but connected to the cytosol. I think there's a lot of stuff. Yeah.
A question from Jane. Does Migrasome help with mitochondrial transfer from one cell to another?
Yeah, we'll try very hard. Like we can get the mitochondrial transfer by the Migrasome to another cell, but we cannot make this transferred mitochondria get like sort of the integrated into another cells like the mitochondrial network. Yeah.
A comment from Gosh. Wonderful work. Is Migrasome formation specific to only migrating cells? Does any other polarized cells generate them like epithelial cells in tissues? Is there a similarity between cytonyms?
Yeah, I think with cytonyl is quite different. Cytonyl is really the field body is active, like the extended out. So for the other cell type, like the now, all like the work is only focused on the migrating. But we definitely say whenever you have the relative, like the movement, for example, the shrinking of the cell, when we find like we never published it, but we find like during the cytokinesis. When the cell really like the dividing, shrinking, you will have a lot of Migrasome-like structure produced.
Yeah.
So I always think like my speculation is probably whenever you have the relative movement of the cell's age, you will see the Migrasome formation.
Yeah.
So that's left a lot of room to study this like the no migratory cell.
Okay. Another question from John Hammer. Many immune cells migrate in vivo with minimal integrin engagement via chimney. I guess in compressed environment, I guess John was talking about bladding amoeboid migration. So yeah. Would Migrasome be formed during such a migration?
Yes, because like I didn't show the data today. So when we imagine like,
we lost you.
Oops.
I guess the internet is
the solid tumor.
Lee, we lost your signal actually. Can you answer this question again, which I think is a really good question from John?
Yeah, I think so. I think this like the order in the very dense packed environment. Could you hear me?
Yeah, now I hear you.
So now like we already get the data from the Migrasome formation inside the spleen, inside the lymph node, and inside the solid tumor. This is all very dense place, and the cell is actually squeezing basically under the pressure from the surrounding cell. This still like to produce quite a lot of Migrasome.
Okay. John said for your information, traction fibers contain mixed polarity actin.
That's good to know.
Yeah. So a question from Xu Yue, amazing. In the cultural condition, how much ratio of the cell can form the Migrasome? I guess the same question as before.
Yeah, depends on what kind of the cell. For example, like our favorite cell is the L9 cell, which is a fibroblast. In that cell, like almost every cell is producing Migrasome.
Yeah. From Marianne, are any protein specific to or with higher expression in cancer cells found in Migrasome?
Yeah, yes. Like the several, like the actually they have this couple paper published, for example, tetraspinin expression like the somehow is correlated with the several cancers like the metastasis ability. So we are not really into this field, but like the I learned something like the people are doing this right now. Yeah.
Can you give some example, Lee?
I cannot exactly remember.
Okay.
Yeah.
All right.
Yeah, but definitely they have the cancer like the cancer specific protein, if I remember correctly. Yeah.
Okay. If no more questions, let me ask a final question. So we talked before between us. You talk about you regard this as organelle, and many people in the field thought of this one special form of extracellular vesicle (EV). What's your take on this?
Yeah, so like so now we have the many evidence to show actually. For example, let me show you these slides. In many cases, migrosome actually didn't detach from the cell. You can see the white area is point. It likes swallow and it shrink back. During this like the expanding stage, what they do is they separate the separate protein site. So it's never become in some of the cell, the migrosome will not detach from the cell. That's just the site of the secretion. So they have the cell autonomous function. The function did not need to become the actual cell vesicle to carry out.
Okay. Yeah.
Yeah.
I agree.
And also, like another thing we are pursuing is the migrosome. From surface of migrosome is highly enriched with the receptor. Now we have evidence in some cases they can become the antenna to receive the information to the cell. Yeah.
You mean to the donor cell actually?
To the cell to generate migrosome. For example, TGF-beta receptor is highly enriched on the migrosome.
Are you talking about the EGF receptor on the surface or in lumen?
Surface of the migrosome,
okay.
On the surface. So it's basically, if you think about it, it's almost like an antenna. With the distance, you can receive the signaling and send back to the cell. Now we still work on this, of course. So like I understand like the naming or like the whether it's called like the EV or is the organelle is not really important. The reason I call that's at the beginning, we already found something migrosome will do is not related, they became the EV.
I agree. Naming is not important actually.
Yeah, exactly.
Yeah. All right. Time is up. I hope everybody really enjoyed like as I did. And let's thank again for Yuli for this wonderful talk. All right.
Thank you very much.
Have a good. Bye, everyone.
Bye.
Thank you.
Thank you, Yihong.